since developed antivirals. So these are small molecules that target essential enzymes that the virus uses to replicate. These antivirals, such as remdesivir, molnupiravir, or nirmatrelvir, are highly potent at suppressing viral replication. However, what we've begun to see is that the virus is now evolving resistance to the antivirals that we're using in the clinic. So nirmatrelvir, for example, has a couple of different ways that the virus has evolved to get around it. One way is to mutate the site that the drug binds. This is a mechanism of direct resistance. So for example, nirmatrelvir targets SARS-CoV-2 protease, and if the drug binding site is mutated, then the drug no longer works. That's one mechanism that you can use to evolve drug resistance, but these proteins are highly conserved and a lot of the sites are required for the virus to replicate. So oftentimes a virus can't just change those positions without losing fitness itself. So what we're beginning to appreciate is there's actually a second route that the virus is using to get around our antiviral arsenal, and that's what's known as indirect resistance. And by indirect resistance, I mean it's essentially improving the viral replicative fitness so that even though you're drugging it, the virus is still making so much of itself that it doesn't care. So for example, there are mutations now in the protease that increase the enzyme activity so much that the drug dosage that we're using of nirmatrelvir isn't as effective as it used to be. Now that was looking at nirmatrelvir, which is the protease inhibitor. However, the drug that is most often used and that most people have gotten is remdesivir. What's interesting about remdesivir is a polymerase inhibitor. And when we look at resistance mutations in NSP12, the polymerase, to remdesivir, they're kind of all over the place. You know, they're all over the protein. So how could mutations in all these different places impact remdesivir activity? The other important thing to note is if you look at the remdesivir binding pocket, which is shown on the bottom, we see no mutations there. So the virus is evolving mutations, but not in the actual binding site of remdesivir. So we wanted to better understand why this was occurring. So in Northwestern Hospital, we began recruiting patients who had a sample taken before they received remdesivir and one that they received after they had remdesivir. So now we have two samples of the same person's virus, but they had a drug in between. And we wanted to see how the virus evolved after the drug was administered in our hospital. And what we saw is we saw that a large number of positions across the genome were increasing in diversity, specifically in the patients who received remdesivir. And what was most interesting about these positions is if we looked at what happened to these positions over the course of the evolutionary history of the virus, we saw a lot of them actually ended up arising in different Omicron sublineages. So what's happening inside the individual human is that the virus is learning how to be better. It's increasing its replicative fitness. And then when these Omicron sublineages came along, a lot of these mutations the virus learned, actually, that makes me a better virus, and they're becoming fixed in the population. I'll skip that one, because I know we're close. We're running low on time. So, evolutionary history of SARS-CoV-2. 2020, we saw increased transmissibility, D614G, lots of virus spreading around. 2021, even more transmissibility, and now it's beginning to gain resistance to monoclonal antibodies that we're using in the clinic. 2022, we saw broad immune escape. We saw vaccine breakthroughs and vaccine resistance. 2023, it diversified even further, and now we're beginning to see antiviral drug resistance. So the question is now in 2024, how is the virus going to continue to evolve in order to evade our immune system and evade our antiviral arsenals? Now, SARS-CoV-2 was the virus that I used to explain how genomic surveillance can really inform how we handle and treat people in the clinic. But that's just one example. So I wanted to very quickly highlight a second virus that was really impacted by this COVID-19 pandemic, and that is RSV. RSV is respiratory syncytial virus. And it usually infects in the US in the fall and then goes away and then comes back and then goes away and then comes back. But when we were protecting ourselves from SARS-CoV-2, and we were wearing masks and everybody was social distancing, RSV started acting very, very strangely. So in 2020, all of a sudden, we didn't see any cases at all. RSV just disappeared. We didn't have anybody sick in the hospital. We didn't have anybody showing up in the clinic. It was super bizarre. But then once everybody started taking their masks off and kids went back to school, suddenly we started seeing kids get sick again in the green line, but they were getting sick in the summer. And RSV is supposed to happen in the winter. And all of a sudden it was around in the summer and it didn't make any sense and things just kind of went crazy. So what exactly happened? Well, one of our students is Stephanie Rios Guzman, who is here teaching one of the workshops that we're working together with Ricardo at CITBM. She has been sequencing RSV to try to understand what happened to the virus when all of a sudden it started acting weird and had different seasonality. And when she sequenced RSV-A, so there are two main subtypes, A and B, when she was sequencing A and comparing before May of 2020 in blue and after May of 2020 in red, she was seeing that the virus more or less looked the same before and after the pandemic. Well, then look at RSV-B. Now, RSV-B, if you look at blue before 2020, it's all clustered together. And if you look at what happened after 2020, the virus all of a sudden has a much different sequence than it did before the pandemic happened. And if you look at exactly what positions in RSV were changing over that time, they're in the RSV fusion protein. What's the fusion protein? It's the RSV version of the spike protein. And which positions were changing? They're positions that are in the antigenic sites that bind to antibodies. So we're seeing the exact same changes in RSV that we were seeing in SARS-CoV-2, where it's evolving to evade the immune system. And Ramon will talk a little bit more about other viruses, but I did quickly want to highlight RSV because I think it's a really cool story. So to recap, the virus is going to continue to diversify and evolve over time. And new variants are going to continue to emerge as long as there's so much virus spreading in the community. Intrahost evolution, zoonotic transmission, recombination, all of these mechanisms are ways that the virus can increase its variability and contribute to the evolution of SARS-CoV-2. Now, the main drivers of this evolution are going to continue to be increased transmission to escape immunity, as well as the emergence of therapeutic resistance. The Omicron variant was really important in this history because it resulted in less severe outcomes. But the impact of these diverse sublineages that are evolving now, we still don't know. It could evolve to become more severe in the future, but we'll have to wait and see. Continued genomic surveillance, especially in regions of the globe that are undersampled, is really important for us to continue to understand and continue to use this information to improve public health. So this is a brief summary of this pipeline that we use to do this type of work. It starts with sample collection. After we have the samples, we sequence them using genotyping. Then we link it back to our clinical data. We then model that data. And then finally, we can report it either in a paper or to a public health entity and validate our findings. Now, this sounds great, but in actuality, it's really hard. And there are a lot of different challenges that inhibit our effectiveness at doing this. And since we're here in part to teach a workshop on genomic surveillance, I did quickly want to highlight just a couple of the challenges that we see. So, for example, with genotyping. So this is sequencing. So what issues do we see when we go to sequence a sample in a clinical specimen? Well, the first thing we could do is we could use rapid tests. So these are PCR-based tests that amplify a portion and say, is the bug in the sample or is it not? These are great, but you really have to know what the sequence of that virus is, or you can't design primers, you can't design probes, it might not work very well. And the ongoing evolution of these viruses mean you constantly have to be updating these tools or they're not going to work. Now, the other approach that we use to assess pathogen genotype is deep sequencing. So this is next generation sequencing like on a nanopore or an Illumina. Now, the amplicon-based approaches are like the ones we used for SARS-CoV-2 or RSV are cheap, but they also have a similar problem where you have to continually make new primers. There are approaches called metagenomic approaches or target capture that enable you to avoid primer amplification, but these are very expensive. Now, the other issue is that even though a specimen might have a bug in it, there might not be enough of it for us to sequence it entirely. So, for example, over the course of an infection in an infected individual, your body is fighting the virus. And so the virus levels are going lower, 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 lower until they're not there. So exactly when you collect your specimen from the person also influences your ability to sequence it. And then finally, this requires highly specialized expertise and equipment. As anybody who's doing genomic surveillance in the room knows, it's a very tricky and long process in order to sequence a specimen successfully. So Ramon is going to talk a little bit more about the different sequencing approaches that we can use and some of the benefits and the drawbacks. We use 3 different techniques or 3 different platforms: the Illumina platform, a Pack Biosciences platform, the Oxford Nanophore platform, and Ramon is going to talk a little bit more about them. But understanding each one of these platforms and the benefits and drawbacks that they have end up being really important for successfully completing the genomic surveillance project that you want to run. And of course, none of this means anything unless you have the bioinformatic expertise to actually interpret your data. So even if you put everything on the sequencer, then you get a whole bunch of data, then what? You have to analyze it and interpret it. So you need computational expertise in pathogen genomics and phylogenetics. And that's one of the things that I'm very excited that we get to be here, because we get to learn from you guys, and hopefully we can teach you guys about some of the tools that each of our institutions are using. And because of that, I do think that these kind of international collaborations and partnerships are really, really important. So just to quickly highlight the importance of this data sharing, data types are often compartmentalized. So what do I mean by that? So the data that I showed you today, some was collected by the public health department, some we collected, some the clinicians collected, and all of these data sets are in the hands of different people. So in order to make conclusions like this, you need to bring all of that data together. Everybody needs to work together to make that happen. However, even if we're able to do that, say, in Chicago, that's only one city. That's only one hospital. How do we expand this to other cities and other hospitals to make sure that what we're looking at isn't just an artifact of the one center that we did it at? And part of this problem, of course, is with what's known as interoperability. So that means that the data I collect might be similar, but not exactly the same as the data you collect. So how do we standardize across our institutions and then end up sharing it? And finally, this has become a huge problem, particularly because it's not equitable. So lower middle income countries have historically had lower rates of genomic surveillance and may lack some of the infrastructure required for the work. And this is really important because it can heavily bias our understanding of outbreaks and of global health. And this becomes particularly problematic when the work gets then outsourced to high-income countries. So really what we want to do is establish equitable strategic partnerships and collaborations to address this issue. So for example, here are all of the SARS-CoV-2 genomes that were deposited in GISAID, which is an international platform to deposit SARS-CoV-2 sequencing information. And the bars are the number of sequences, and they're colored by the income of the country they came from. And if you look at the color of these bars, they're almost all red. They're coming from high-income countries like the US or the UK. And so that meant that we ended up learning a lot more about what was happening in those countries than we were at other parts of the globe. So helping establish these kind of collaborations and partnerships is not only important for improving our public health networks, but also for future pandemic preparedness. And so one of the exciting things that we're doing here with Ricardo at CITBM is teaching and being part of a workshop to help develop and teach some of the next generation sequencings that we're using. And so we have an ongoing workshop on nanopore sequencing. I took this picture yesterday because I thought it was really cool. And that's it. The last thing I want to highlight is that we've come a long way, and that all of these problems, there are solutions to all of them. They're difficult to implement. They can be expensive. They require a lot of work and a lot of trust and a lot of collaboration. But their solutions are there. And I think it's up to all of us who are doing this type of work to invest in them and continue to improve these pipelines so we can be better prepared when the next pandemic comes along. So with that, I did want to quickly thank the amazing team that did this work at the Center for Pathogen Genomics. A huge thank you to our hosts here, as well as to all of our funding sources. And with that, I'll turn it over to Ramon.
Gracias, muchas gracias por la presentación. No sé si vamos a dar pase a Ramón. Si tenemos preguntas, esperamos para el final para no retrasar un poco. Tenemos ciertos retrasos. Entonces, vamos viendo. Muy interesante lo que nos ha presentado. A continuación, vamos a tener la presentación de Ramón Lorenzo Redondo, quien nos va a complementar con la parte de bioinformática y otras técnicas relacionadas a estos sistemas de vigilancia genómica, ¿no? Como indicaba el doctor Hartwig, siempre hay desafíos que afrontar en este tipo de trabajo. Sin embargo, con el trabajo colaborativo que se está forjando desde ahora, esperamos nosotros también contribuir a estos desafíos globales. Bien, no sé si ya estamos listos para poder iniciar. Just for the question, maybe later for, si hay algunas preguntas, por favor. De repente, en tanto, Ramón se va preparando la presentación. ¿Tenemos alguna pregunta del auditorio? Puede ser en español. A ver, adelante.
When you guys collected the residual diagnostic samples, do you get any consent from the participants or?
That's a really excellent question. So when you're working with clinical samples, there are two major types of study. One is called a retrospective sample. These are samples that were collected for a different purpose that already exist and that you can essentially reuse for your research. Now at Northwestern, anybody that comes into our hospital and takes a diagnostic test, they sign an automatic consent form to release those specimens in case they're needed for public health needs. So we were able to establish a protocol to collect those samples and de-identify the information so that we cannot link it back to an individual's name or address or other identifying information. However, that was only part of our samples, because the other way to run these studies is what's known as prospective sampling. So that is essentially you tell the person that you want to collect a sample specifically for a research project that you are running. They run through the protocol with you, they will sign a consent form, and then they will give you the exact specimen that you're looking for. So when we were sequencing people over the course of time in our hospital, we had asked them to give both nasal swabs as well as blood samples periodically over their hospital stay, and that was a prospective study. So we do a little bit of both, but it's an excellent question.
Okay, if there's no more questions, I think we can go with Ramón now, please. Gracias. Do you have a... ¿Necesitas un puntero?
Creo que
está el puntero. Ups, sí está bien. Me parece que ya sea un ratito ahí se ha puesto en pantalla completa. Any other questions? Sí, también en español, ¿eh? Puede ser, sí, no hay dudas, pero... Sí, es importante lo que mencionaba, ¿no? Si bien es cierto, se está trabajando, working with residual samples. Do you still need like some ethical clearance for doing this kind of analysis? Because some of the samples, so clinical collecting samples, that you usually do in the routine practice in the lab. So with the reminder that you go to this repository for doing this research, I think you need an additional ethical clearance, right?
Yeah, so there are a number of different ethical hurdles that you need to consider when you're performing these types of studies. We talked about the two different types of sampling methods that you could use, but regardless, you need to get clearance from the appropriate university ethical boards. So at Northwestern, they're called the Northwestern Institutional Review Board, or the IRB. And they will review the ethical nature of your study and license you to perform it or not. If they have concerns, they'll also work with you to help improve it such that nobody's privacy is at risk and that everybody's being treated, equitably. It's a difficult thing to do, this type of research, for exactly that purpose. And clinical trial work is even more complicated than that. But again, it's really important. So we do appreciate all of the work that the ethicists do on our team to ensure that we're doing everything, as best that we can. Yeah.
Lo puedo decir en español. I can speak in Spanish. Yeah. Okay. Because it's so difficult. Me parece un trabajo muy interesante. Es a big, es una data impresionante de toda la ciudad de Chicago. Y que la pregunta exacta es, ¿qué le parece a usted sobre esa concentración que hubo en la publicación, sobre todo en el COVID, cuando el COVID era un ejemplo de enfermedades que podía haberse transmitido de animales a... Zoonotic, yeah, zoonotic, disease is zoonotic, because there are a lot disease zoonotic and a potential diseases for humans and a pandemic, but in six years, approximately 6 years, because it's hasta ahora, the investigation is totally important to COVID, focus in COVID, not in other zoonotic diseases. So what is your opinions about that?
Yeah, no, and I think that's an incredibly important point. COVID was really interesting from a virologist standpoint because finally the world was paying attention to a zoonotic disease. And there's so many of these diseases that jump into the human population very regularly. So in the last 20 years, we've seen over 200 new viruses enter the human population. 200 of them. It's just mind-boggling that so many people don't pay attention to this because we've seen these really important viruses enter the population all the time. HIV is an excellent example. If we had not even then, we struggled. SARS-CoV-2, there were massive problems in our response to how we handled it. And so how we better inform the public and educate people on how we can prevent and prepare for these pandemics, I think is essential. But the challenge is immense. So for example, bats alone have over 2,000 known viruses across 28 different viral families. And that's just looking at one species of bat. Then you look at all of the different species that are out there and the number of different viruses that there are, trying to even catalog them and determine how many there are is difficult. And then trying to assess which ones might be problematic is important. So I'm really impressed by these global pandemic preparedness plans that are now beginning to be established. And essentially the idea behind these plans is that we can prepare in two really important ways. One, we can develop diagnostics for a bunch of different families of viruses so that they're ready to go in case something does jump. Because we remember very early in the pandemic, diagnostics were a huge backup. We were all sitting at home and we couldn't even get tested to see whether or not we had the virus or not. So they're trying to come up with this technology now. The second major thing that they're doing is they're investing in vaccines and preclinical trial of antiviral drugs so that we can be better prepared again if these families of viruses jump in. One of the problems that we had early was that, especially in the US, we were prepared for an influenza pandemic. And then it was a coronavirus pandemic. So we didn't have a diagnostic, we didn't have a vaccine, we didn't have an antiviral, and it's just our ability to predict which virus is going to jump and which one is going to be next has proven a little futile. So being best prepared across all these different viral families is really important. And I think it's a great question.
Thank you so much for your presentation. I was amazed because you create the data, but also you have the tools to make so happen, right? So as students, here you will find like a bunch of studies looking for different kind of things. Somebody is working with bioinformatics. In my case, I'm working with recombinant proteins, but I find it a ways to design the protein as well. So as a student, looking for find a place where those ideas can happen. So you can describe your laboratory, which tool do you have, maybe the chances that you can do protein modeling as well, or somebody's working with the design of the proteins inside your group. Thank you.
Yeah, and I think that's an important thing to recognize, and I failed to do it, and I apologize for that. All of the work from my lab that I showed was done by graduate students. So Stephanie Rios Guzman, who's here, Ted Ling Hu, they did an amazing amount of work in order to come up with the conclusions that they did. And part of the reason is that we have a really supportive team within our Center of Pathogen Genomics. When it comes to modeling, I'm terrible. I can't code, it looks like a disaster. But Ramon is an absolute expert. And so we rely on and help each other in a very collaborative sense. So I think when you're looking for the best place to do your graduate studies and how you can take your work to the next level, try to find a support network that can really enable you to do a variety of different things. So if you have a PI or a professor that's really supportive of you, that's also willing to collaborate with a lot of other people, and they get along together and they're supportive of the work and the work that graduate students do, I think that's the most important thing, even more important than the technology or the resources available. Because even if they don't have it, if they're willing to reach out and work together, they'll be able to gain access to that for you.
Gracias. Ahora sí.
Sí, sí. No, muchas gracias. Bueno, darles las gracias otra vez por esta invitación y por la oportunidad. Voy a intentar darla. Hace tiempo que no doy una charla en español, pero espero que, perdónenme si no me sale bien. Yes. Entonces, bueno, pues la idea es la siguiente. Hemos visto lo que se puede hacer, cómo podemos estudiar los patógenos. En particular vamos a hablar mucho más de los virus, pero esto se aplica a bacterias, a todo lo que queramos estudiar, o incluso procariotas. Entonces, la idea es ir un poco a las razones o a los procesos por los que vemos estas consecuencias en los virus. A ver, bien. Normalmente, sobre todo con estudiantes, siempre empiezo con esta figura. No les voy a preguntar, pero normalmente les pregunto ¿qué es esto? Sí, mejor me voy a poner aquí, a ver, y voy a le voy a quitar esto por si acaso. Entonces, esto que tengo aquí es la única figura que existe en el libro El origen de las especies de Darwin. Es un libro un poco arduo de leer porque no hay ningún soporte visual, pero es uno de los libros más importantes para biología, medicina, para prácticamente todas las áreas relacionadas con las ciencias de la vida. Y lo que ocurre es que él con colaboradores pensó en esta manera de representar el proceso que estaba identificando de continua evolución por medio de una especie de árboles como linajes familiares en las que de unos individuos surgen otros y otros y otros a lo largo del proceso de evolución. Y aquí, y relacionado un poco con la pregunta anterior, da igual lo que estudiemos, si es una proteína, si es un virus, si es cáncer, si es... Lo que creo que siempre tenemos que tener en cuenta en el campo de la biología es que todo ha sido el resultado de un proceso evolutivo. Este proceso evolutivo puede ser selectivo, puede ser incluso aleatorio, pero de todas maneras hay que verlo siempre en esta perspectiva de evolución. Ok, esto va siempre así, no sé. Maybe things that have some videos might be the thing. So yeah, maybe you need to advance it a little. I think the heavier. Entonces, sí, dale uno. Push one more if you can. Yeah. Este es un vídeo estúpido de unos dibujos, pero básicamente lo que quiero que entendamos y por cómo podemos estudiar estos procesos es que todos los procesos evolutivos tienen sistemas complejos de evolución. Uno de los típicos es estos carreras armamentísticas evolucionarias en las que sistemas complejos en los que hay, por ejemplo, un virus y un hospedador, una presa y un depredador, procesos simbióticos, todos no son estáticos, son procesos continuos y dinámicos para establecer el mismo sistema, que al final es el virus hospedador. Es decir, todo va evolucionando. Y en este vídeo estúpido es una cosa